Exonate is a small UK-based ophthalmology company. We have expertise in alternative splicing, and we're developing small molecules for the treatment of retinal diseases, specifically focusing on a splicing factor kinase called SRPK1. The leadership team is here today. It's myself, I'm co-founder and the chief executive, and Loic Lullier, who's my chief operating officer. We're backed by a group of investors from the UK and from Australia who've supported us this far through our phase 1 study and who continue to support us to the future. And we are about to raise our Series A. We're very proud of our clinical advisory board. These people have been helpful to us from the beginning. We have representatives from the US, from Europe, and from Australia. We also have a clinical development team who are individuals who have been working in this area for many, many years and have helped to guide us through phase 1 and the planning of our phase 2. So what do? We're trying to make eye drops for the treatment of neovascular retinal disease. We have a group of small proprietary molecules which we have manufactured to be selective, to be potent, and to reach the back of the eye under their own merit rather than being dragged there by something. We finished our phase 1 study. We got a positive signal. We inhibit SRPK1, which is splicing factor kinases that causes rebalancing of VEGF isoforms. I don't think I need to explain to this audience about diabetic macular edema. However, this is a very large market, not just in terms of the number of patients, but also the dollar value of it. However, that does not mean there's not room for improvement. About 40% of patients show an inadequate response. In most areas of the globe, those patients with early and mild disease are not eligible for anti-VEGF injections. And of those patients who are actually offered an appointment time, about 30% of them fail to show up for the appointment. We did our clinical trial in Australia, 48 subjects. We chose patients with mild to moderate disease simply because we felt it was ethical. We didn't know if our drops would work or not. So the average CMT was 320, and they all had a BCVA of above 69 letters. As in most clinical trials, our primary endpoints were safety and tolerability. We then looked at a secondary one for PK, and then because we were in patients, we had exploratory endpoints trying to see was there effect at the back of the eye. We did 3 doses, 0.5, 1, and 1.5. 1.5 people were treated for 3 months. So we met our primary endpoints, this safe and well-tolerated molecule. We had no serious adverse events, and it didn't cause any problems to the eye. Our PK profile was good. We had low systemic availability and no accumulation. I wanted to show you a picture of the back of the eye. Now, this is only one patient. I completely accept an N of 1 is not science, but if you look at it, this is day 8, day 29, day 57, and by the time we get to 85, 3 months later, the edema has gone. This is just one patient that worked. There were about 320 to start and 260 to finish. Now, when we accumulated all the data, there was no significant difference between patients on placebo and those on active treatment. However, I have to tell you, it was not powered for efficacy. We needed 200 patients to be statistically relevant, and we had 35. However, there was a statistically significant difference between people at baseline and day 85. Not huge, but these people didn't have very boggy retinas. And in those who were on placebo, as you can see, it was all over the place. We therefore looked at what would happen if you took this cohort of patients and used anti-VEGF agents on them. And as you can see from here, patients with very mild disease, you don't get a big response. And the black dot, I think the black dot is us. No, we're the blue dot. The blue dot shows that we are on par with other anti-VEGF agents in this group. However, if you take patients with more severe disease, you do actually get a bigger drop. So that's what we're planning to do in phase 2. I wanted to show you some images of our fluorine angiographics. If you look, all of these patients were responders. The top 5 pictures are the screening visit, and the bottom are day 85. And with the eye of faith, it does appear that there is an effect on vascular leakage. We therefore sent the blinded data to an independent third party and said, could you grade this and analyze it for us and just send us back the results? If you look at the data going from screening to day 8, there's no difference in patients between placebo and active, which is absolutely fine. The mechanism of action would not really expect that. However, when you look from day 0 to day 85, there is a significant difference. Those patients on active treatment had a decrease in vascular leakage, and those on placebo had an increase. So we think that this is well worth looking at and taking forward, because from our exploratory endpoints in mild patients, not statistically relevant, we did see a reduction in CMT, and we did see a reduction in vascular leakage. We think EXN407 is a good molecule to take into phase 2. We have seen high exposure in the retina in animals. We've got low systemic availability data in humans. It was safe, it was not toxic, and we're going to take it forward. Thank you very much.